It is a pleasure to have Dr. Bi as our speaker today. Okay. I'll just give a very brief introduction of him. So he graduated from Brandeis University, 2012, and afterwards he worked at Syracuse University from 12 to 15. And now he is at the Rockefeller University. And he is a specialist in soft matter and biophysics, theoretical part. So, the floor is yours. And thank you for coming. With a token from our department, we thank you. Okay, so this is a small token gift from the department. Okay, the floor is yours.
Thank you for the invitation. I really enjoyed my visit here. So I'd like to tell you about my recent and ongoing work in looking at the mechanics of dense biological tissues. So a lot of work in the field of biomechanics and biophysics have focused on how a single cell moves and how a single cell shape is generated with the different mechanisms. But here, when you have a dense collection of cells, when they're really crowded together, cell interaction becomes highly important. And it's not just how a single cell interacts with the substrate, but how a cell can mitigate such a tight environment by squeezing itself past other neighbors. So we'll look into the mechanics of tissues like this. This is one example. A confocal microscopy image, confocal slice of a 3D tissue aggregate that was taken in zebrafish explant. So these dense environments for cells actually occur quite a lot in many biological processes. If you think about wound healing, this is called a wound healing scratch assay. So you have initially a full tissue that covers the entire substrate in 2D, and you can take a needle and scratch the wound. So you make a gap. Initially, the cells retract, but after some time, they can actually collectively move to heal the wound gap. And this is, I believe, MDCK cells, which is a common cell line that forms a nice monolayer when plated in culture. And dense tissue migration also occurs in cancer metastasis. So when cancer cells become tumorous within an otherwise healthy tissue, they're usually dormant. But the bad news is when they start to migrate. So when they start to migrate, cancer cells will have to move through this very dense environment that is formed by healthy cells and try to make their way into the bloodstream, eventually trying to get more and more nutrients. Once they enter the bloodstream, it's essentially like the highway in your system, and it makes it really easy to get to other organs and spread to other parts of the body. And also during developmental processes, this is the axis elongation inside a fruit fly embryo during the developmental stage. There will be a lot of cell rearrangements in these dense environments. And you see this process of zooming into a fruit fly embryo. You see a lot of local rearrangements happening in order to form, in this case, the head-to-tail axes for the fruit fly. And this is done this is done when the cell number isn't really changing. So cells are not dividing. All this motility is due to the tensions that you get by certain myosin concentrations. So if I have time in the end, I'll talk a little bit about my work on this. But also during development, cells, different types of cells, for example, cells that are going to form different organs, will undergo sort of sorting processes, which is also collectively moving towards the parts of the body that they need to form. And the red dots you see here are one type of the nucleus of one type of the cell. And you don't actually get to see the other, but they're really densely packed. So in this environment, they can actually collectively sort out according to which type will eventually form the skin and the muscle, and the other type goes sort of inside the body and forms the internal organs. So as a physicist, the question you might ask, what is the material property of a dense tissue? And simple tests like this, a simple yet very interesting tests have been done to probe mechanical property of a tissue aggregate. So this is a similar system to the video in the first slide. It's a zebrafish embryo explant consisting of about 5,000 embryonic cells. And if you take this tissue aggregate and put it under a parallel plate, what happens if you compress on it? So the answer really depends on what is the time scale of perturbing the tissue. So the first video will show a compressing for seconds and let it go. So it's a very short time scale behavior, and you see that the tissue aggregate basically bounces back to the original shape rather quickly. Maybe you can infer from this it's very elastic in its nature. Okay? But the same tissue aggregate if you take two of these, put them side by side, if you give them long enough time, let's say on the order of hours or days, they can actually merge very much like two liquid droplets. So there will be very viscous behavior at this long time scale. And you can apparently infer, you can look at the shape that these tissue aggregates form. There's a lot of tension effects. So this looks really like two liquid droplets, or let's say two oil droplets in water merging. So the tissue mechanical response really depends on the time scale you're looking, the time scale of perturbing it. And this is not surprising in physics. We just call this viscoelasticity. So what is the origin of this viscoelasticity? And this can be answered by looking at the microscopic dynamics of individual cells. So if you can track the cells, which was done in this paper by Eva Maria Schultz, at the short time scale, when the macroscopic mechanical response is elastic, essentially the cells are very much trapped or caged by their neighbors. So within that short time scale, they can't go very far. And if you look at a single cell motion, it's very much just rattling in that cage, but not able to uncage and go to a new location. But given long enough time, they actually visit multiple cages. So they're able to hop between these cages formed by their neighbors, eventually diffuse. So if you look at mean square displacement as a function of lag time, eventually it goes to a slope of 1, which means diffusive motion. So the consequence of being crowded together means that you have these cage-like effects, which ultimately leads to viscoelasticity. So is there any way we can predict or control this kind of behavior in dense tissues? Can we have a framework that describes when it transitions from a fluid-like state to a solid-like state? And precisely what tunes the viscoelasticity is the question that I'd like to answer. So the type of models that people have used to describe these dense tissues initially have basically fall into this class so-called self-propelled particle model. Essentially, you represent in a very sort of coarse-grained and on a simple level each cell as just an isotropic particle. So a disk in 2D, a spherical body in 3D, and they interact with some interaction that's usually short range. So you have some interaction upon contact usually. And you can model the cell motility by saying that there's a polarization associated with each cell, and the cell will tend to exert motility force along the polarization vector. And you can write down different rules for how these polarization vectors interact with each other, as well as how it evolves in time. But the important thing to take away from these type of models is that cells are represented as sort of isotropically shaped bodies that have short-range interactions. And a lot of work has been produced with these type of models, and it does answer a lot of questions. And the intuition that by using these models can be actually answered or sort of inspired by the work that's done with particle models that are not biological, that are, for example, colloidal particles. So people actually look at the phase diagram at the dense limit for colloidal particles and ask if this is a good picture, perhaps also for biological tissues. And this is work done by Dave Waite's group at Harvard, where they essentially study the phase diagram, the phase behavior of dense colloids that have attractive interaction. And the axes are really easy to understand. So changing density means essentially changing the packing density of the particles. And at some critical density, they will undergo what's so-called a jamming transition. And essentially means that each particle is forced to touch other particles and it has no place to move. So this is sort of where you get to a random closed packing. And then increasing the density will just make it more and more solid-like. So you can certainly get a fluid-like, go from a fluid-like state to a solid-like state by just increasing density. And that density-driven transition is often called the jamming transition. And then temperature can certainly change the material property. So think about temperature so the jam state is basically the solid-like state. So within this surface, imagine all the states are solid-like. By increasing density, you can go out of the solid-like state by essentially melting the material. So you can have enough thermal fluctuations that make the particles move around. But adhesion for these particles essentially means that upon increasing the adhesion interaction, and for particles it's just the attractiveness of the particles, you can get a gelation transition, where the particles will form a percolating gel network that goes to system size, and you can get a rigidity, you can get mechanical rigidity by going this route. Okay? So this is very much the intuition for particles. Whereas increasing adhesion means you get a solid behavior. And applied shear is just straightforward. Applying enough amount of mechanical deformation can also make the material flow. And so biologists took this picture and said, let's try to model cells with particles. And can we use this picture to describe the mechanics of dense tissues if you were to model them with particles? And this is a hypothesis based on the previous phase diagram put forward by biologists at the Harvard School of Public Health. And essentially, you still have the density-driven transition. So at some critical density, you go from fluid to solid. And adhesion is that intuition from particles, right? So increasing adhesion means you get solid-like behavior. Replacing the mechanical load or the stress is the motility of individual cells. So here, because it's more biological relevant to think about single cell motility as the control parameter, this can also make the tissue flow. But if you look at this picture, the previous examples of actual biological collective migration are all occurring at very dense limit. That is that they're so densely packed, there's no room or gap between cells, right? So they really all look like this picture. They're all very dense, sitting in deep, in terms of this phase diagram, they're all sitting densely in here. But yet in those systems, let me show this video again, they're really at this packing fraction of 1, which means they take up the whole volume or area in 2D. There's no gaps between them, and biologists call it the confluent limit. Yet there's still a lot of motility going on, and if you observe, this is a 3D picture, so you see this cell that can actually go out of the plane, right? So this is a confocal scan of a 3D system. It can go from this plane to the next. So there's still a lot of rearrangement and migration, even in this dense environment. So the picture, the phase diagram that was put forward here really doesn't explain fully why you can get collective cell motility and flow and viscous behavior at this high density limit, because the last phase diagram would just say this should be a solid, it shouldn't be rearranging or moving around at all. So we need something new to add to the previous picture. And truly, there are many examples where a density-driven transition is not quite possible. And here's one example that I'll go into more detail, but just to show you, these are the same tissue culture looked at different dates. And this is taken from the bronchial epithelium of human patients. I'll tell you more detail about this later, but all you should take away from this is that initially, the tissue is basically flowing around. So this is a 2D monolayer. Cells are moving very much like a fluid within the tissue monolayer. So you can use all of the tools from condensed matter to characterize whether the tissue is a fluid or solid, and you would say this is very much a viscous fluid. On day 6, looking at the same tissue with the same number of cells, so division doesn't happen between these states, and essentially it slowed down significantly, okay? And then on day 10, it's basically completely slowed down. It's a quiescent static state where the cells are really not able to go anywhere. So this is very much a transition from a fluid-like state to a solid state, while the density is completely constant and at the dense limit. So how do we explain such a system? And the type of model that I have used is not new, has been around for a little more than a decade, and it was originally started by these Japanese biomechanical engineers, so-called the Vertex Model. So if you think about cells that form a monolayer in 2D, right? Essentially, the Vertex Model considers interactions between cells that generates the shape they can take in this 2D monolayer. And let's just go through briefly what effects will be captured by this model. So if you think about just two cells, right? Two cells sticking with each other, what generates the shape that you see? Well, first of all, the reason that they are sticking with each other are due to adhesion molecules sitting at the edge between the cells. And adhesion molecules are usually some type of cadherin molecule or a catenin molecule. And the effect here is basically the more of these adhesion molecules you have, there's more of an energetic tendency to share a longer boundary, right? So what this is doing is if you're able to control the amount of adhesion molecules, the shared boundary length will be longer and longer. And on the other hand, there's also the presence of a high concentration of actin-myosin gel network that basically form the cortex around the cell. And what this is actin fibers that are in the periphery of the cell with myosin motors walking along the actin fibers. And this generates a contractile tension, basically, around the perimeter of the cell. So if you compare these two terms, essentially the contractility of the cortex gives you something like a tension effect, whereas the cell-cell adhesion counteracts that tension. So this will tend to shrink the cell, whereas this actually wants to grow the perimeter of the cell. Now, also there's elasticity associated with the cell's shape, so we take that into account. And also finally, a cell usually resists fluid exchange. So you can consider the cell as a 3D object that conserves its volume. So this will be taken into account in writing down the interaction. And just putting this together, you arrive at a very simple mechanical energy functional for a single cell. And so essentially I'm writing down mechanical energy associated with the 3D incompressibility, which translates to, in 2D, just a quadratic area term in the energy with a preferred area. And the preferred area is dependent on the actual volume of the cell as well as adhesion between one cell and its neighbors. So this actually basically comes from the 3D incompressibility as well as the resistance to any height fluctuations within the monolayer. So this is saying that the cell doesn't want to fluctuate very much away from the rest of the cell heights. And the second term, quadratic in the perimeter, essentially captures to first order the elastic effects associated with the actin myosin cortex and also the internal cytoskeleton's elasticity. And finally, last term, you have a gamma times the perimeter. So gamma here is an effective tension that is controlled by two things, right? As I mentioned earlier, it's an effective tension that is generated by the amount of adhesion you have between the cells and the amount of cortical tension. So adhesion molecules will actually decrease this gamma, whereas cortical tension increases the gamma. So assuming that all cells within the tissue have the same mechanical property, that is that they all have the same parameters within this energy functional, you can non-dimensionalize this and write a total mechanical energy functional for the entire tissue. And after all, you just end up with these two parameters by completing the square with the perimeter. So you get a perimeter minus a preferred or target perimeter P0, which depends on basically this gamma, and a R, which tells you the relative strength of the perimeter term and the area term. So it's very, very simple model, and writing it this way helps you to maybe think about the geometric meaning of this. So geometrically, there's a preferred perimeter as well as a preferred area. So P0 and R will be my parameters for this model. And thinking about mechanical property of the tissue, of course you can think about, this is simulation, of course you can do anything you like. You might be tempted to just say, let's test the bulk, the shear modulus of the tissue to get a mechanical behavior. And that's true, you can do that. But what's more relevant is actually to think about how cells move in this dense tissue. Basically, if you think about this dense tissue where it's densely packed, so there's no gaps, and cells really can't go out of the layer. And if you don't allow cell division or cell death, the only way, it should be easy to convince yourself, the only way for a cell to move from point A to point B is to execute a bunch of these local neighbor rearrangements. And from the foam language, it's called the T1 transition. Essentially involves this red cell, it involves 4 cells, and this red cell initially not touching the other red cell can squeeze past the constriction formed by these 2 green cells and make a new contact with the red cell. So they make a neighbor swap and end up in the new state. So this is just a demonstration of a series of T1 transitions. It's not a true animation, just to demonstrate if I do by hand a bunch of T1 transitions, that's the way you can move around within the tissue. So basic idea here, this is the basic unit of motion. And what I want to capture the mechanical property of the tissue with is the amount of work the cell needs to put in order to make these local rearrangements. So this is the limiting factor for migrating within a tissue like this. So what I do is I induce these T1 transitions by hand, and I measure quasi-statically, meaning that at every step of making this T1 transition, that I minimize the energy functional and calculate what is the energy as a function of doing this T1 transition. And typically this is what a energy trace as a function of this path looks like. So initially you start at some state, and by this cell trying to squeeze past, you go to a cusp, and then making the transition at the cusp, you can relax to a new state, either at a higher energy or lower energy, depending on the disorder or the local topology in the system. But this, you can think of the energy barrier here, right, basically from the initial state to the cusp, as the amount of work the cell has to do, an estimate of the amount of work the cell has to do in order to make this basic unit migration. So, and I can do this for all possible transition paths within a tissue. I can make all the possible T1 transitions and measure the energy barriers. And what I find, this is a schematic of the energy landscape, is that imagine you're going from one state, which is initially sitting at minimum. By making the T1 transition, you go to a different minimum, and by overcoming a local energy barrier. So by doing all of the possible T1 transitions, you get some idea of what the energy barriers look like. And the interesting thing is the energy barrier or the energy landscape dependence on the parameter P0, right? That's the effective tension or the target perimeter due to adhesion versus cortical tension. And it seems that as you increase P0, the energy landscape actually changes in that the energy barriers are lowered and lowered. And at some point, at a particular value of P0, you essentially get a flat energy landscape. So what this means mechanically is that if you're in this state, it takes a lot of work. The cell needs to really expend a lot of energy in order to go from one state to another. But if you're in a state like this, the cell can just put in a small amount of work to go from one state to another, and eventually basically can just flow because it's completely effortless. So if I plot the average energy barrier for a tissue, averaging over all the possible transitions, and plot it as a function of P0, this is what I was talking about. So the average energy barrier actually vanishes at some point. So you can think of the role of this R parameter essentially changing the sharpness or the overall scale of the decrease towards this point. But basically at every R, it's undergoing this trend. So these are for several R values. And what this should remind you of is that if you think of this as an order parameter characterizing the mechanical behavior of the whole tissue, then the mechanical rigidity is really being lost as you go towards this point. So it's a change or transition in the rigidity in the mechanical property. And if you think of this furthermore as an order parameter, and perhaps this P0 as something that can tune the distance to this transition, and this might have an analog in other phase transitions. And you can think of this R value essentially giving you the strength or controlling the amount of fluctuations near that transition. So based on those ideas, people, of course, to characterize second order phase transitions can do something like this with the order parameter. And this you can do in the simplest sense with the Ising model. You can basically take your order parameter and assume that in the system there's a critical point. In this case, phi is the density, and there's a critical transition at this phi c, assuming that phi minus phi c controls the distance to the critical point. And you can write down a scaling ansatz by assuming that there's a length scale that controls everything near this critical point. And if the scaling ansatz works, actually you should be able to collapse the data for your order parameter onto two branches. And if you look at granular systems, jammed and unjammed systems, and where the order parameter is really viscosity, so in this case viscosity can collapse onto two branches, where here is a viscosity that basically will tend to diverge. And here for the fluid-like state, you have some finite viscosity, meaning that indeed the scaling ansatz works, and confirming that it has the features of the second order phase transition. And this can be also done with randomly cross-linked fiber networks, whereas here the order parameter is the shear modulus, and the distance to the critical point is controlled by how many random bonds sort of needed for the percolating rigidity percolation transition. And you can again collapse the data onto two branches. One is a floppy state where the shear modulus eventually vanishes, but also a rigid state on the other branch, giving you the mechanically rigid side of the transition. So taking those ideas, can we cast my data of the energy barrier in terms of the scaling analysis? And the advantage of that will be to show that, look at what kind of universality classes it belongs to, if it is indeed a second order phase transition, and how different is it from these other types of transitions that you have already seen. So it involves writing down this critical scaling onset, and my hypothesis is that the critical point occurs at P0 star, so where that energy barrier goes to nearly zero, but I don't know how to figure that out accurately, so I can make a scaling onset like this, saying that P0 minus P0 star controls the distance to critical point, so this acts something like T minus TC in, let's say, Ising Model counterpart. And then when you write it like this, it should scale onto two branches, one for the rigid side and one universal scaling for the soft or floppy side. And R essentially acts like the magnetic field in Ising Model. So I can take this data and apply this, where my beta, delta, and the form of the function are the fitting, are done through fitting. And indeed, if you fit it to the correct exponent, you can exactly collapse all of this data onto the two branches, where one is P less than P0 star, collapse onto the rigid branch, giving you basically finite energy barriers. So that's the mechanically rigid. And the other fluid branch that has a vanishingly small energy barrier. So this really confirms that there is a diverging length scale in the system, and it is a proper second order phase transition in terms of the rigidity of the tissue. Okay? And through this process, we can figure out the exponent, but also more importantly is where the phase transition occurs. It looks like 3.8. But I can figure out that exactly P0 star is at 3.813. So I'll tell you about why those numbers actually have any meaning. But here is the very simple phase diagram. So the distance to the critical point is tuned by P0. And on the left side, if you have P0 less than P0 star, which is when the tension is stronger, right? So you have a rigid tissue. And this is a typical morphology taken from simulation. But if you increase P0, either due to decreased cortical tension or increased cell-cell adhesion, you can actually get a very mechanically soft tissue. Okay? So think back to the picture with particles, where the more attractive particles are, they actually become more rigid. But here, the more adhesive cells are, they actually become softer. So it's sort of maybe a counterintuitive result, but this is because cells are really not modeled well by isotropically shaped particles, because cells, after all, are deformable objects, and adhesion actually has a different effect for these deformable polygonal objects. So the transition occurs at this P0 star, and it's 3.813. And actually it has some geometric meaning. So if I think about P0 as the preferred perimeter or target perimeter for the cell, then I can correspond these certain values to geometric shapes. And in fact, where a transition occurs corresponds exactly to a polygon of 5 sides, a pentagon. And if you decrease the P0, essentially the lowest P0 value that has a correspondence to a geometric shape is the perimeter of a circle. So that has the least perimeter for a given area, right? And you can increase the amount of perimeter, and the largest one corresponding to a shape is triangle. And it's really interesting that the transition is actually to a fourth decimal point agrees to that of a perimeter of a pentagon. So let me give you an explanation why I think that is. This is an argument I am making that is based on very mean field counting arguments. So, I think it is correct, but I don't want to say that this is a proof. Okay. So if you have any ideas, please make any suggestions that you can. So this is very simple counting argument based on isostatic conjecture. And this is originally, of course, due to Maxwell, James Clerk Maxwell, in counting the number of constraints you have in the system versus the number of degrees of freedom, right? So if you have the exact match between these two numbers, then you begin to have the possibility of having solutions in the system. And this is basically based on that isostatic counting. If you consider for a cellular structure, how many number of degrees of freedom are there? Well, you just count the number of vertices in this network. So so the red dots represent the vertices, and the lines are the edges in the network. So if V is the total number of vertices, right? So for each vertex, I have 2 degrees of freedom in 2D. I can move in X or Y. So I have 2V total number of degrees of freedom. And for each cell, I have the mechanical equilibrium constraint, meaning that for each cell, for a rigid tissue, for each cell, it cannot, all the forces acting on the cell have to be balanced in the rigid state, right? So for each cell, if there are n number of cells, then for each cell, I can write down 3 equations telling you the mechanical force and torque balance. So 2 equations for force balance and 1 equation for torque balance. That's where 3n comes from. And very simply, for mechanical equilibrium solution to exist, the number of degrees of freedom has to be at least greater or equal to the number of constraints. So 2b greater or equal to 3n. And coupled with a couple of constraints that come from topology. So this is just Euler's counting for this network. Essentially, the number of vertices minus number of edges plus number of cells have to be equal to 0 for a flat space, right? So I'm on a flat space without any curvature. And then also each edge is shared by 2 cells, right? So if I count up the total number of neighbors each cell has, right? And Z is the average number of neighbors per cell. So for this cell, it's a hexagon. It has 6, but in a disordered lattice or a tissue, it can certainly vary. So this Z times the total number of cells divided by 2 has to be commensurate with the total number of edges. So this is just a statement saying that each edge is shared by 2 cells. Just taking all this and solving for the inequality, exactly you get Z equal to 5. And this says that in the isostatic mean field limit, the mechanical equilibrium constraints match up with the number of degrees of freedom exactly at 5. And this is where I make the leap to say that perhaps this 5 in mean field means energetically I would need to prefer a perimeter of that of a pentagon, 3.812. So that's my argument, and I haven't been able to disprove it, but this is not meant to be proof at all. It could be wrong. Please ask any questions if you have any idea. So, what am I doing on time? Ooh. So let me skip this. So far, I've talked about the equilibrium mechanical property of a tissue. I've talked about minimizing energy to calculate this work that the cell needs to do. But it's really not a dynamic model, right? But you will look at the videos from before and say cells are really moving around. Where is cell motility? So next, I'd like to talk about my implementation of putting in cell motility together with these into this vertex-based model for cells. And this is a new model that I developed that actually takes the best of both worlds from particle-based models and shape-based models and combine them to simulate cell motility. So the way that I put in cell motility itself, it's not so interesting. It's really not so new. It's essentially saying that I start with a bunch of points, and on each point I give it a polarization. That's the cell polarization, which means that the cell will tend to go in that direction due to some motile self-propelling force. And that polarization vector is n, and that n vector you can write down as complicated dynamics as you like for that, but here I go with the simplest, essentially saying the cell has a vector, and that vector will have some random rotational diffusion. Okay, that vector will rotate randomly given this time scale dr. So you can, of course, put in correlation between the vectors, between the cell and its neighboring cells, and people do that, but here I want to do the simplest version of cell motility. But the next part is quite new. It's essentially taking this particles, this collection of points, and you can get the cell shapes by doing a Voronoi tessellation. So I use a Voronoi tessellation to represent the shapes of these individual cells, and then I calculate the effective force between this cell and its neighboring cells based on the energy functional that I showed you earlier. And the advantage of this is not quite like any particle-based model where the interaction only depends on intercellular distances. It really depends on the interaction between, let's say, this cell and this cell will depend on where all their neighbors are. So in that sense, it's a multicellular interaction, multi-body interaction that's not exactly just a local interaction that's between, that's parallel. So putting this together, the interaction force due to the Vertex model and the motility self-propelling force, I'm simulating the overdamped equation of motion for each cell. And so this is due to the interaction between the cells, and this is due to the motility. So here, just to emphasize that the forces here are inherently multicellular and cannot be expressed as pairwise interactions. Okay, so now we basically have some additional parameters. So before we had P0, which controls that rigidity transition, and now I have a V0, which is the amount of motility force, and dr, which is the diffusion constant for the rotation of each cell. Okay, so let's look at a phase diagram of how those additional parameters will change the picture. So before, we know that there is a rigidity transition at 3.8, around 3.81, when there's no cell motility. So what happens if you increase cell motility? If I'm slightly above 3.81, which puts me in the floppy or the soft state, and just give it a little bit of cell motility, then you see the cells are starting to float, which means it's really easy to overcome whatever energy barriers or the lack of any energy barriers in the state, and it's really easy to create a fluid state. And these different colors just track several cells, and just to show that they actually move certain distances over a long time. So this will be sort of a viscoelastic state that's comparable to the experiment. And if you go at the same amount of cell motility, but if you stay in the rigid state, applying this amount of cell motility really doesn't get the tissue to float. So it's very much still solid-like, even though it's maybe jiggling around a little bit. But the cells don't have enough cell propulsion to go over those energy barriers. It's very much staying like a solid. But at the same P0 value, if you increase cell motility, you can get a very fluid state. It's essentially has a very gets into a diffusive regime very quickly, and it's a very sort of fluid, much more fluid-like state than what you have here. So there must be some change, either as you change V0 and P0, you can cross from a fluid-like state to a solid-like state. And I'd like to figure out exactly where that happens, and if it hits that point. So I drew this line because I know the result, but basically I want to characterize where the transition happens. So this will be a rigidity transition without any cell motility. And now this transition from this fluid-like state to a state like this is basically a glass transition, right? So the definition for that is, you go from a disordered fluid state to a disordered solid state. Here, although you're not quite changing the temperature of the system, but it's another source of energy input via cell motility. So the kind of measure that you can characterize fluid versus solid with, there are many of them. Here I simply choose the diffusivity of the cells. So if you look at the mean square displacement of individual cells, in the fluid state, usually you will have some initially holistic regime, and then it tends to plateau, which means that that's the relaxation time involved with cage breaking, and eventually it becomes diffusive. But in some other branches, for some other value of P0, it can plateau on forever, so it never becomes diffusive. So you can extract basically this final value of the mean square displacement, scale it by the amount of time it takes to reach there, and define a self-diffusivity. Basically, if you run the simulation for a long time, and if ds is a finite number, then you are in a fluid state. If ds is 0, then you are stuck in the solid state. So characterizing with these dynamical measures with the self-diffusivity, you can break the phase diagram in terms of V0 and P0 into two phases. One is the fluid and one is the solid. And interestingly, the transition, there's a line of glass transition that originates from where the rigidity transition was. Okay? And I can show you a few pictures. So, so the cells in the fluid states actually look like this. And if you plot some cell tracks, they actually are indeed diffusive. And there's something called dynamical heterogeneity emerging, which simply means that as you get closer and closer to a glass transition, the length scales and of the displacement of your cells get highly correlated. So in time as well as in space. So essentially you see the formation of these highly correlated swirl structures. And the size of the swirl will increase and actually become system size as you go to the glass transition. And in the solid state, you can look at some cell tracks as well, and they're really caged locally, so they're not going anywhere. So experimentally, we try to think about what kind of measures you can suggest for experiments to measure that will predict such a transition. And if you think about the snapshots that I showed you before, the cell shapes actually look quite different. So if we go back here. So in the fluid state, you see the cells tend to have mostly larger perimeter, and in the solid state, they seem to be more compact, right? So can we, derive a simple shape-based order parameter to tell which phase you are in? And so I just started to look at this very simple parameter, which is dimensionless. You just take the perimeter of the cell, scale it by the square root of the area, and average it over the whole system. And it turns out that this parameter exactly, when this parameter is equal to 3.813, it overlays exactly at where that glass transition boundary is. Okay? So this means that, so by the way, so the value, this is the control parameter P0, right? This is what you control. And at 3.813, you undergo the rigidity transition, but this is what you measure. So it seems that when Q is measured to be this value, then you're exactly sitting at the glass transition boundary. And then if Q is greater than this number, then it's at a glassy or fluid, it's at a fluid state. If you're inside, it's less than 3.81, it corresponds to a solid state. Okay? So this should be really useful for experimentalists because you can imagine measuring something like diffusion will need to take very long dynamical ranges, right? You need to take the data for a long time in order to determine whether it's really diffusing or not. But here, I'm suggesting you can simply look at a few snapshots and look at the shapes of the cells and be able to tell what its mechanical property is. So I should just briefly, the effect of this dr parameter essentially tells you how persistent the motion of each cell is. So if dr is 0, that means, so dr is essentially the rotational diffusion, right? So if it's very low, that means the cell basically undergoes super persistent motion. If dr is high, that means it's constantly changing its heading. So this is more like in the Brownian regime, this is in a more persistent type of motion. And what I'm showing here is that for all values of dr, they indeed will shift where the glass transition happened, but ultimately all emerge from where the rigidity transition is. So this just says that no matter how you explore the energy landscape, which has this rigidity transition, they're ultimately controlled by this phase transition. So let me skip that, but I want to go to the experimental verification of the work here. And that is the videos I showed earlier. This is cells taken from the bronchial epithelium and plated in culture. Okay, and these cells in a healthy person will have cilia growing on top. So the function, the physiological function for having these cilia is usually to trap mucus and to move mucus along in the airway so that they don't block. So if you imagine by either coughing, some mechanical perturbation, or external chemical stimulus, you can injure the airway epithelium. And it seems the response to that injury is that it will go into a fluid-like state. And so responding to the injury is initially to go into a fluid-like state, and then in a few days, it will repair that injury and go from a fluid-like state to a more solid-like state, or it's becoming quiescent in a few days. And this is all done without the cells changing in their number density. So this is what happens in a healthy person. If you take the cells from the healthy person and plate it in culture, over the number of less than 2 weeks, it basically stops moving. And the idea is that maybe when it stops moving, cilia can grow from the tissue properly and be able to trap mucus as if it were in vivo. But if you look at the cell from an asthmatic donor, people with asthma, so I didn't show the data from day 3, but on day 6, it's moving still very much in the tissue. So these are the velocity, the displacement vectors of the cells. And on day 10, it's essentially still moving a lot around. Compared to the day 10 from a healthy person, the asthmatic donor is still not able to stop, basically not able to complete its injury repair. And on day 14, it's significantly slowed down, but there's still activity. There's still some motion within the tissue. So they did a lot of measurements here by looking at the velocity, looking at whether it's diffusing or not. But experimentally, they really don't have the convenience of looking at the tissue for many days. So ideally here, if you could take a snapshot and be able to say at which stage it reaches from a fluid state to a solid state, that I think would be very useful. And that's where we came in. So we suggested to these experimentalists at the Harvard School of Public Health to look at this shape-based order parameter Q. Okay, so from their dynamical data, by looking at the velocity of the cells, they would say that for a healthy person, on day 6, it's almost jammed, meaning that it's almost stopped moving. And on day 8, it's totally jammed. It's basically no motion at all, quiescent tissue. But for the asthmatic person, it takes 14 days to almost to be as slowed down as the day 6 here. And that's from their dynamical data, right? Our prediction from the model would say that exactly at the 3.813, above that you should be fluid-like, and below that should be solid-like. So we asked them to measure the individual cell shapes, and I should say that the data is not the cleanest because they currently don't yet have a good image analysis algorithm. So they actually had a student clicking on cell borders to calculate cell areas and cell perimeters. But ideally, in the future, we'll improve that and get more statistics. But here, if you look at the Q from these experiments, for the healthy tissue, it really gets quite close to where the transition is. And it exactly gets a trend. As you grow it for more days, you get to the jamming or the solidification threshold. But for people with asthma, it has a similar decreasing trend, but it doesn't quite get there, showing that it's not fully solidified yet. So this prediction seems to really work well for these systems, which don't change in density, yet undergoes a transition from a fluid to a solid state. Okay, so I'm almost running out of time, and my conclusion is that liquid to solid transitions in tissues is pretty ubiquitous and even occurs in the very dense confluent limit, important to many, many biological processes. And we find that there is a rigidity transition that is controlled by cortical tension versus adhesion as opposed to just particle density. And there's actually a glass transition line that emerges from that rigidity transition if you add cell motility. And based on that, I can predict a cell shape index, this Q number based on perimeter and area of the cell to distinguish between fluid phase and the solid phase. And this is purely based on static structural properties, which is a huge advantage over dynamical measures. So finally, I want to acknowledge the people I worked with. Some of this work was done when I was a postdoc at Syracuse, and this excellent graduate student worked with me on implementing the cell motility into the Vertex model, and it was not so trivial to calculate the forces the effective forces due to the energy applied on each cell's Voronoi tessellation. And my postdoc mentors helped me greatly, Christina Marchetti and Lisa Manning at Syracuse. And the experiments were done at the Harvard School of Public Health in Jeffrey Fredberg's lab with these postdocs taking measures of cell motility and growing the tissues. So I'd like to thank all these folks. And that is the conclusion of my talk, and thank you so much for your attention. We have time for some questions.
Could you show your transparency where you have the elastic energy in terms of perimeter square? Yeah. So you said if you complete the square, this energy can be written in terms of P minus P0. But Kp and gamma are both positive, aren't they?
So they don't. So gamma doesn't have to be positive,
Yeah.
because gamma is controlled by these two things.
So the tension would have to outstrip the adhesion in order for gamma to be negative. Is that right?
So adhesion, the other way around.
Other way.
Adhesion will have to really be increased, or tension is really downregulated. The tricky thing with biology is that these effects are never independent of each other. So if you put in adhesion molecules, you're not just decreasing the tension due to adhesion, you're also in changing the tension itself. You're also changing the cortex tension. So you're downregulating the other thing. And overall, you can write this down as an effective tension that is due to these effects.
What about finite size effects here? So it seems like you have perfect data collapse with this huge large number of cells, or you have to be restored.
So the data collapse I showed here is at a fairly large system, but I did do finite size effects, and there's finite size scaling and technogenics going on that. Yeah, not in terms of, I didn't do finite size effects in terms of this local energy barrier measure, but I actually did finite size scaling of the shear modulus itself, which turned out to be a slightly better quantity if you're doing critical scaling.
Did you want experimental side?
Yes.
Last view.
Last view.
Yeah. Seems like the error very large.
Yes, that's right.
So are they still significantly different?
Yeah, I think the error is larger because, this is a way of testing the theory, but I think ideally it will be better to have an algorithm that can do this systematically. So this is clicking on a small amount of cells, maybe hundreds, given a tissue that's maybe 1,000 cells large. So that is one source of error. And another source of error is that I think perhaps one should consider a tissue where the individual, let's say the individual P0 values are not singular, right? So not all cells have exactly the same single cell property. There must be a distribution of these, which can also wash out or make the transition less sharp. And that could be the source of these fluctuations as well. But I think the trend is pretty convincing, and also the trend correlates, that doesn't show, but the trends also correlate very well with something that they're more sure about, which is the velocity measurement in terms of whether the tissue is moving and how it slows down. So these shape measurements exactly correlate to those velocity measurements.
Have you looked at Penrose tilings?
Quasi-crystals?
Yes, I mean, the one pattern, because they're based on, 5-sided symmetries and they don't repeat, and your one looks just like it. Back up a couple slides.
Yeah, so Penrose tilings have a quasi-crystalline structure, but these are truly amorphous. But it would be nice to look at the mechanical property of a Penrose tiling under this energy function. I haven't done that yet.
You talk about the adhesion and motility, right? To me, I would likely think these two are highly correlated, right? Because they must have kind of the same source, the surface of the cell, right? The characteristic of the cell surface that will govern those. So how correlated are they?
So they are correlated to some extent, but the mechanism for adherence here is usually, say, due to cation molecules that, like PCAD, NCAD, that sit at the junction. And usually cell motility are due to focal adhesion. So adhesion sites that cells form with the substrate. So the cells can grab onto the substrate and then pull themselves. So I think there is certainly, these two are not completely independent, but I should say here, maybe, I should say that I'm not able to, for example, for that experiment, I'm not able to precisely say where the experiment, which path it takes in that phase diagram. So all I can say is that if I take this for example, so by just looking at the cell shape, I know that it's somehow approaching this glass transition boundary, but it could be taking that path, or it could be taking a combination of this path, because decreasing motility means changing adhesion and tension, or maybe it's doing something like that. So we're working together with these experimentalists to see what is precisely the mechanism, and can you tune those more independently.
It seems to me that you assume in the 2D model, you seem to assume the total area, the overall area is fixed, right? But in reality, if we add salt solution, and change the shape, overall area.
Salt solution meaning that you affect the liquid exchange.
Right.
Yeah, so that's another interesting area which has not been explored here. Certainly there's recent work showing that cells can actually fluctuate in their overall volume quite a bit in some cell lines.
In response to the area
Or-
change of environmental change,
Right.
temperature, salt solution. Can you model that?
That's something I can model, for example, in response to calcium changes, which open up different channels that helps you to exchange fluids. And those things are sort of something that I'd like to do are in my future plans.
Yeah, another question for me is, you seem to assume pretty uniform cells.
Yes,
Can you model different types of cells? Like
Yes.
some are very small, some are very big.
that's right. So that's an excellent question, and I think a little later I'll go into some of my current work where I do assume a difference in the single cell properties, and when you mix them up, a lot of interesting things can happen.
Okay, let's send our speaker again.